welcome to today's Lab 8 Automation Masterclass, Biological Barriers to Automation. We're going to be covering essentially all of those considerations that are specific to biology with the kind of samples that we might look to be working with in biology. So I'm Marcus Kasheta, I'm co-founder and CSO at Synthace, and I'm going to be your host. And I'm really excited to introduce our very own Dr. Stephen Brown, who's a senior research scientist here at Synthace. Steve has a PhD in industrial biotechnology. Like me, he's a DOE evangelist, and he's got over 7 years of experience with lab automation. So he's ideal to tell us a bit about some of the technicalities that are involved in getting stuff to work in biology. Steve, I'll hand it over to you.
Thanks very much, Marcus. And yeah, good afternoon, everyone. Hope everyone's well. I'd like to just give an overview of the series so far, which can be viewed on demand. And this is linked by the Synthace website. So go there for all the on-demand stuff that you can watch at your leisure. So previously, Daniel introduced us to 3 types of automation used in the life sciences, namely flexible, specialized, and integrated. And he showed associated user cases for each of these. Nathan then looked at some key liquid types and how, based on their properties, you can adapt liquid classes for a device to improve both accuracy and precision. And most recently, Anne and Luke took us through the process to implement automation within your laboratory and how to tackle a couple of common challenges to the adoption process, namely, I can do things faster by hand and that the learning curve is too steep. And so that brings us to today's webinar on biological barriers to lab automation. And straight off the bat, I'd like to start with an apology for this title, as it's not the best, other than the pleasant alliteration. I think the term barriers puts a negative taint on a very achievable endeavour of a scientist being able to run automated biological experiments in the lab. Maybe it should have been considerations when running experiments using laboratory automation, although this isn't very pithy, not a very good title either. Anyway, let's move on. And building upon the previous webinars in this series, I will present some practical recommendations when running different types of automated biological experiments with a focus on liquids and environmental impacts that are fairly commonplace during a biologist's research activities. So what is meant by biological barriers within the context of laboratory automation? So these images highlight some of the biological liquids under consideration when using automation in the lab. In general, there are 3 main types of macromolecules used in biological experiments, namely cells, protein, and DNA. I found this research paper where the authors were looking at the impact of these biological liquids on viscosity during the course of a fermentation process. I'm highlighting it here as, from a chemical engineering point of view, liquids containing cells, proteins, and DNA all generally impact the viscosity of a liquid. The authors showed a linear relationship between the cell concentration and viscosity. And interesting, these large error bars here for the higher cell concentrations were thought to be caused by cell aggregation from centrifugation and resuspension during the sample preparation steps. So they'd have expected it to be better had the preparation been better. The authors also showed a relationship between protein concentration and viscosity. Here they noted that E. coli contains a vast range of proteins, as we all know, and although they differ from BSA, they believed it is rather the total concentration of protein that determines this impact on viscosity. So here they were using BSA as a proxy for total protein concentration. And finally, they showed a linear relationship between DNA concentration and viscosity. So in summary, all these different types of biological liquids shown increase the viscosity of a liquid. However, I wanted to show you that this impact is not always linear. It can be a bit more complex. And this complexity can impact our strategies that we might employ to handle biological liquids for an automated protocol. In this paper here, the measured viscosity values were similar for lower protein concentrations, as you can see here. However, there is an exponential trend observed for the higher protein concentrations. The authors used HPSEC, a type of chromatography, to look at the aggregation profile of recombinant albumin, looking at whether the protein was in either the monomeric, dimeric, or trimeric configurations. You can see that the increased viscosity at the higher protein concentrations coincides with the aggregation of the protein in solution. These observations highlight that things can be complex. Let's now take a look at handling viscous liquids in general, with a view to help our understanding when moving our biological molecules of interest using laboratory automation. So let's first take a look at the general properties of viscous liquids. These properties of viscous liquids will be familiar to most scientists, as they're likely to work with viscous liquids from day to day. First, we've got resistance to flow. So when a force is applied to a viscous liquid, it flows slowly in response. You can see this when you're manually pipetting and you press the plunger to expel the liquid, and you can see the flow of the liquid is delayed, leaving the tip. High cohesion is where the liquid interacts with itself, sticking to itself and making it resistant to changing in shape. The image shows here this droplet as an example at the end of the tip. High adhesion is also a general property of viscous liquids, where they can stick to other materials such as plasticware, like the tips and microtype plates that we use for lab automation. You can see this droplet halfway up the tip here, adhering to the side of it. And the viscosity of a liquid is temperature dependent, with the viscosity decreasing with increasing temperatures. So with those properties of viscous liquids in mind, let's take a look at some strategies to improve the accuracy of pipetting them during an automated workflow. One thing we can do to improve the accuracy of viscous liquid transfers is to choose an automated device which utilizes positive displacement, such as the Dragonfly here. The delivery of a liquid using positive displacement pipetting means that the action of dispensing is not impacted by the compression of air or system liquid, which may be used to move the liquid of interest. The operation can handle higher pressures caused by the viscous liquid's resistance to flow. And this means it can deliver a constant flow rate of the liquid. Let me run this video showing you how this works and how the cone on this plunger is in direct contact with the liquid. So there you go, you can see the plunger moving down, the cone in direct contact, and as it moves, it directly moves the liquid out the tip. You can also use reverse pipetting to improve the accuracy of transferring viscous liquids. This is where you take up more volume than is required for the desired transfer. And this can be achieved for liquid handling automation by specifying an over-aspiration volume. If you operate at fast dispense speeds, using only the exact volume in the tip that you require to transfer, then due to the adhesive properties of a viscous liquid, droplets can remain in the tip, leading to under-pipetting. An over-aspiration volume, taking up some more liquid than is required, can help alleviate this. You can also make different selections of tips for an automated protocol, as well as vary the set points for a liquid class for the automated device. You can choose wide bore tips to reduce the resistance to flow based on the tip. You can change the material of the tip to counteract the adhesion of the liquid to the tip surface. Just in general, a liquid class defines the set points of parameters used to control aspiration, dispensing, and mixing during pipetting actions on an automated liquid handler. When handling viscous liquids, both liquid level detection and liquid level following can be enabled to allow the pipette tip to track the liquid during aspiration and dispensing steps. In the video, you can see how the tip finds the level of the liquid and then tracks just below the surface during aspiration and dispensing. In this way, you're preventing excess liquid from sticking to the outside of the tip, which in turn may then be dispensed to your destination well, impacting accuracy and precision. Controlling the flow rate to make it more consistent, like with the previous example using positive displacement pipetting, can also improve the accuracy of the transfer. Additionally, reducing the flow rate and slowing the speed at which the viscous liquid travels in the pipette helps for both aspiration and dispensing. I think you may have experienced aspirating something like 50% glycerol too quickly, and the resistance to flow that means when you take up a bit of air when you remove the tip from the source liquid. You can also slow the speed at which the tip is removed from the liquid. This is useful when you can't use liquid level detection and following. You give time by moving out slowly to allow the liquid on the outside of the tip to be removed by the liquid in the source. And you can have a time delay after the piston is moved during either an aspiration or dispensing step, whilst the tip is either still inside the source liquid or at the desired dispensing height. This time delay allows the liquid to complete its movement either into or out of the tip, preventing potential underpipetting during the transfer. The final consideration in this section is for you to think about your automated protocol as a whole. Think about whether you can dilute a potentially viscous stock so that it's no longer a viscous stock. And think about the potential of allowing stocks obtained from cold storage to get to room temperature before using them on the automated experiment. This would likely reduce the viscosity and make them easier to handle. The next slide is looking at how we can measure accuracy and precision of a liquid transfers and can be applied to your liquid of interest, not just viscous liquids. In Nathan's webinar, he introduced the RTEL MVS system that we have in our laboratory at Synthace. It is used to determine the accuracy and precision of liquid transfers. I have an example here where we've used it to measure the accuracy and precision of these different types of liquids on an automated liquid handler. Here you can see the precision of the different volume transfers from 0.2 microliters to 18 microliters. You can see the BSA solution in orange is particularly problematic and has higher precision compared to the liquid types. And this was in large in part due to it creating air bubbles being prevalent in the destination well after it was dispensed using the specific liquid class. I think the key thing to take from this is that it's useful to understand the limitations of your device and associated liquid class, as you can make a decision on the volumes to be used for your automated protocol. Without further optimization work here, we would likely ensure the minimum transfer volume during the protocol using these liquids would be 4 microliters, as this is where the device performs within good levels of precision. Nathan also highlighted an example in his webinar where the liquid class was optimized for the handling of DNA during an automated qPCR protocol. DNA and master mix are combined before performing a very sensitive qPCR assay. The work he showed highlighted how DOE could be applied to the set points of a liquid class to optimize the transfer of DNA, improving precision. These results show the top 5 best performing liquid classes. Initially, the experiment was conducted using a base liquid class for the transfer of the master mix, which was kept the same whilst the optimization of the DNA liquid class was performed. They then wondered whether the assay would perform better if it used the same optimized liquid class for both the DNA and master mix transfers. This set of new data shows that for the majority of the liquid classes, that the precision of the automated qPCR assay was indeed improved. The key takeaway from this is that liquid classes can be transferred between different liquid types. So if a liquid class that performs well, maybe you can transfer its use to perform the liquid handling for another liquid type. But at the very least, it might be a good starting point for subsequent optimization experiments. So now we're going to look at an assay example where we wanted to assess the impact of handling cells using automation. The cell assay we were interested in was the cytotoxicity assay. Cytotoxicity assays are typically used to assess cell survival in response to some stimulus, such as, for example, different compounds. In this instance, we wanted to assess the effectiveness of different CAR molecules in targeting T cells to kill cancer cells. To do this, the different cell effector batches need to express the same level of CAR molecules on their surface in order to allow for a direct comparison between them once they're incubated with the target cell. We wanted to transfer the currently manually executed assay to one that uses automation and a liquid handler. The following is going to be a flow diagram of how the cytotoxicity assay is set up. To set up this assay, we first need to transfer a set number of target cancer cells into the final assay plate, pre-mixing to ensure that the cells are well mixed and that we can achieve an accurate desired target concentration. Next, we want to ensure all the effector cell batches are transduced with CAR molecules with the same efficiency. To do this, cells are normalized with non-transduced, I'm going to have to read this, peripheral blood mononuclear cells to the same transduction efficiency. And only then are they added to the final assay plate at different ratios compared to the target cell. This plate is then incubated for 2 to 3 days, and after which the percentage survival of the target cells is established. So the results I will now show you are from the work to look at these pre and post mixing steps that you see about, seeing how an automated cytotoxicity assay compares to a manually executed one. When trying to automate this protocol, it was imperative to ensure that we were transferring accurate numbers of cells for each condition. So we assessed varying pre-mix and post-mix cycles to get the target cell number correct, and to do this with the minimal impact on cell viability. The maximum speeds possible with the device used were 37 microliters per second, and this was used for all the pipetting steps. And what we did here was observe the immediate effect on cell viability straight after the creation of the assay plate. You can see here the requirement to do some mixing to ensure an accurate transfer of the total cell numbers for the assay. This doesn't have any pre or post mixing and is a lower concentration compared to the other. And we thought this could be due to cell settling, which is something we'll talk about a little bit further on in the talk. Summarizing the data, there seemed to be no difference to the cell number and percentage cell viability when comparing the manual and automated experiments, providing they're well mixed. This was an initial assessment of the target cells, as we thought the automation may drastically impact cell viability immediately. However, we needed to do further work to test within the context of the assay time window of 2 to 3 days to see if this was true. So next we ran a mock cytotoxicity assay. We set up the mock cytotoxicity assay as previously shown, with the exception that the T cells were transduced with GFP with varying efficiencies. The transduced effector cells were then normalized to the same percentage GFP and then mixed with target cancer cells. This is done so we go through the assay but won't be killing the target with the impact of having CAR molecules present. This was then followed by incubation for up to 2 days to assess the overall automated protocol, comparing it to the results of a manually executed protocol. So you can see here the different affected GFP cell batches using either the manual in black or the automated experiments in blue. This is the combined percent viability of the 2 cell types. And we can see after we get this initial drop in viability for the automated protocol, things pick up to a similar level compared to the manual protocol by day 2. So this result wasn't too concerning. It was when we assessed the total viable cell number for each cell type that it became clear that the cells handling using automation were in fact affected and didn't proliferate as readily. The cells were viable, but they just weren't growing as well. This mock assay had shown us that there was that with the current automated protocol as it stood, the impact on cell growth would likely skew the results of the real assay, so not good. And so we reduced the pipetting speed for the device from 37 to 10 microliters per second for all aspiration, dispense and mixing steps. This is the data for the cytotoxicity assay using the CAR T cells. The NT data is you can see it here in black. The NT data is the negative control for the assay, so it just uses the target cells. And you can see how after the reduction in pipetting speed, now there are similar results between the manual and automated protocols. This was a good result and meant that we could now use the automated cytotoxicity assay in our research, and achieved by slowing pipetting speeds down. The key takeaways about the development of this automated assay is that you have to choose your metric to assess the assay correctly. In this case, the viability alone didn't show us the impact of pipetting at the initial higher speeds. It was looking at the impact on cell number that showed we needed to optimize things. And the second thing is we needed to assess over the course of the 2 to 3 days, matching the actual assay timeframes. In the cytotoxicity assay, we saw how mixing was important to transfer the desired concentration of cells to assay plate. So now look at how you might assess mixing for your automated device. So let's take a look at an assay that we have used to understand how, it's more of a method, I guess, how efficient our mixing strategy is on an automated liquid handler. As with the assessment of accuracy and precision for the viscous liquids discussed earlier, we're using the RTEL system to quantify the volume of the liquid being transferred. The first thing is to pipette a volume of buffer that we are going to mix our quantifiable dye into. For this example, we then take 8 microliters of the quantifiable red dye and dispense it to the bottom of the well. Next, we can vary how we mix the sample in the sample dye into the buffer to assess different set points on mixing efficiency. We can change the mix volume, and this is linked to something else that we track, which is the proportion of total volume mixed. So if the volume in the wells is 100 microliters and we set the mix volume to 50 microliters for 2 mix cycles, then the proportion of the total volume mixed is 1. We can change the flow rate used during the mix step, as well as the number of cycles a mix action is performed. We can adjust the liquid type that we want to add the red dye to, so we can create things like 20 gram per litre BSA sample dye or DNA sample dye for testing. We can adjust the height at which the mixing will take place. This is usually referenced from the bottom of the well, so it may be 1 millimetre above the bottom of the well or 8 millimetres above the bottom of the well, somewhere nearer the centre of the liquid column. Then we aspirate from this well that has undergone a certain mixing strategy, half of its total volume, transferring in this case 45 microliters of the upper portion of the initial well's contents to a second plate. Plate 1 is then taken to the RTEL system where it undergoes vigorous shaking to ensure that liquid is homogeneously, that word, mixed before measuring the total volume of the red sample dye in that well. A concentration gradient metric is created for the results of the RTEL measurement, where we take the volume measured in plate 1 and divide this by half the initial sample dye transfer volume and then subtract 1. So in this example, say we had perfect mixing, we would measure 4 microliters using the RTEL. And the mean transfer volume is 4, that's what we'd expect. So 4 divided by 4 is 1, and then subtract the 1 is 0. And this 0 represents perfect mixing. If we had no mixing and all the dye remained at the bottom of the well in plate 1, after the transfer step, we would measure 8 microliters using the RTEL in plate 1. The mean transfer remains the same, so 8 divided by 4 is 2, subtracted by 1 is 1, and 1 represents no mixing. And so that is the concentration gradient metric we use to assess these different mixing strategies. It's also the hardest part of the talk. So let's look at some more results. On the y-axis, you can see this concentration gradient metric where 0 is well mixed and where you would like the mixing strategy to perform. On the x-axis, we have proportion of total volume mixed, where total volume mixed number, where 4 cycles of 50 microliter mixed volumes would equal 2 proportions of the total volume mixed. This is the same as if it were 8 cycles of 25 microliter mixed volumes. On the left side, we see the results for 70% glycerol sample dye, and on the right, a water-like sample dye. The colours represent the set points for the post-mix volumes used in the different mixing strategies. The data is a combination of two runs where we ran something first and then realised we needed to increase the proportion of the total volume mix above 2 to see better mixing, something that was quite unexpected. So with no mixing strategy, no mixes at all, the results are close to 1, showing poor mixing. This is what we'd expect. We thought some of these aqueous results that show the well was properly mixed may be down to diffusion of the water-like sample dye over the course of the experiment, and we'll come to that in a couple of slides' time. Looking at the data for the proportion of total volume mixed being 2, here and here, we can really see the impact of increasing the post-mix volume on improving how well things are mixed. Increasing post-mix volume increases how well things are mixed. And we can also see how the 70% glycerol is harder to mix than the aqueous sample. What was surprising from the results in this example is that you need 11 cycles of 50 microliter post-mix volumes to really improve the mixing of the 70% glycerol. It's quite surprising. This is similar to the previous data, but the colours here indicate different pipetting speeds using during the mixing. Again, a concentration gradient of 0 is indicative of good mixing. And here we can see, particularly for the 70% glycerol sample dye, the impact of mixing rate on promoting better mixing. So if you were to take this data forward for subsequent mixing liquid classes, you would ensure that they're greater than 75 microliters per second flow rate to promote the better mixing rate. Here, the sample dye was added to the buffer and then either transferred to the second plate relatively quickly within 20 minutes or more slowly after around the 50-minute mark. There was no mixing of these wells, and you can see for the aqueous liquids how the sample dye is diffusing and almost becoming homogeneous after the 50-minute mark. So this is just to highlight that diffusion does happen and that sometimes no mixing is required for certain liquid types. It will diffuse over time. So we wanted to find out if the method I've just shown you to assess proper mixing might also be applied to cell settling. These are a list of the things that can affect cell settling. There's quite a few that can impact it. And so we really wanted to create a method to be able to assess it. And if we can optimize any automated mixing strategies to ensure accurate transfer of cells. This is the method that we just showed you. And we wondered if we could swap the Artel volume measurement for the original method to using the cell counter, measuring the cells per mil in both plate 1 and plate 2 after the transfer steps. We thought if this worked, we could be looking at different automated mixing strategies over different periods of time to see how well we could transfer cells. And apologies again, but this is very preliminary data, but maybe we can use this method. On the y-axis, again, you see the concentration gradient, and on the x is the time at which the top 45 microliters of the transfer is transferred to the second plate. The 3 sections are showing either where there's no mixing or 3 or 6 cycles of pre-mixing before the transfer. The colours highlight the position at which the tip was during mixing, either 1 millimeter above the well bottom, the blue, or 8 millimeters above the well bottom, closer to the center of the liquid column. With no pre-mixing, we can see how over time we get settling of the cells and more heterogeneity in our liquid. And when you do a pre-mix, you seem to improve how homogeneous things are. Additionally, there may be some evidence that mixing from the center of the well, the red data, also improves how homogeneous things are. The next section is looking at the potential for contamination during automated experiments. Before we get into an example, let's look at some of the things that can lead to droplet and aerosol formation. So after an aspiration from a source well, droplets can form on the end of the tip. This may be due to the adhesive or cohesive properties of the liquid of interest. To alleviate this, you can use liquid level detection and liquid level following to prevent coating of more of this liquid on the outside of the tip. Additionally, adding a delay after aspiration can give time for any material on the outside of the tip to return to the source well. Here we show the incorporation of a trailing air gap with some air aspirated after the source volume. This can prevent that droplet being present on the end of the tip, which is a concern if it is present when you move the device head moves across the deck. It may fall off in some random well. This comment is that you want to minimize the height that the orifice of the tip is above the top of the well. This can occur if you define your plates to be shorter than they actually are, or if you specify a Z height for dispensing as such. You want to minimize the likelihood of the dispensing action into the desired well, potentially contaminating neighboring wells by having the tip dispense within the well. That's what makes a bit of sense. Additionally, after dispensing, if there is a droplet on the side, a side touch action could be formed where the tip is moved to the side of the wall to remove the droplet. If this is used, it is best not to reuse the tip, as this contaminant can contaminate the source due to what's been previously in the destination well. So let me show you an example of a side touch using the Opentron's liquid handling device, where it actually touches all four sides of the square well after dispensing. Boop. There we go. This diagram on the left is showing aerosol formation at tips, something that typically occurs when pipetting solvents in particular. However, liquid in the headspace of a tip in an uncontrolled manner can go into the pipetting channel of the liquid handler itself, and this can occur for all liquid types, not just solvents, particularly if you're using very fast pipetting speeds, or if you're going to the well bottom to do an aspiration and the plate hasn't been defined correctly. The tip can sit on the bottom of the plastic and create a vacuum. When the tip moves up, all of a sudden you get a great rush of liquid into the tip, and this can contaminate the head. Liquid in the pipetting channel of a device can lead to cross-contamination, as well as require servicing or replacement of the channel. You could use positive displacement devices to alleviate this, such as the Dragonfly dispenser shown earlier, where the cone is in direct contact with the liquid. One of the key things for other liquid handling devices is the use of filter tips to protect against this, providing a barrier between the working volume of the tip and the channel of the device. Filter tips are not foolproof though, and scripting a greater volume for your transfer than the working volume of the tip can lead to something like this. Here you can see that liquid can indeed travel through some filters. That's not good. So now on to an example where we wanted to see if we could use automated liquid handling to create different media compositions to optimize a cell culture project. The thing was that we wanted to use the Tecan Evo in the main lab, where we also culture bacteria and yeast, and this device was not in a sterile hood. The work sounds risky, I know. The work would look at transferring media on the Tecan Evo and then seeing if any contamination becomes apparent over the next few days. The first step that we decided was to wipe all the surfaces of the device using 70% ethanol. We then ran the following experiment, which took about 30 minutes in total, and we ran it twice, once using sterile standard tips and once using sterile filter tips. The source wells all contain the same media. I'm going to make this preview. And this preview of the experiment shows that we were mimicking the transfer of mixing cells and media together before performing a serial dilution. So something similar to what we wanted to do for the cell culture project, but in this case, only using the same media. It's going to go again. Okay. After we generate these plates, either with the sterile filter tips or the sterile standard tips, the plates were covered and then incubated for 37 degrees, whilst periodically measuring absorbance at 600 nanometers to see if there was any potential contamination of the media. This is the absorbance data from a control plate that used the same media but was pipetted manually and in a biological safety cabinet. The media by itself was the negative control, giving us the reading for the absorbance of the media with no contamination, with the readings being between 0.06 and 0.09. The positive controls were added by added E. coli into the media, and so these were the impacts on absorption if things did become contaminated and turbid, and here we're measuring around about 0.5. This is the plate created by automated experiment using sterile filter tips. I have to apologise for the third time because of the colouring of this output from the plate reader colours things relative to the readings for this specific plate. But looking at the values, we can see that they're comparable to the negative controls, showing no contamination with filter tips. This one that's highlighted as very high is actually the same value as the negative control. So no contamination when we use sterile filter tips on deck. These are the readings from the automated experiment using standard tips or non-filtered tips. And we could see there was some contamination in wells comparable to that of the positive control. Some are okay, but a few are contaminated though. And this final table for this section shows the results from a plate where the media was dispensed manually in a biological safety cabinet and then was left on the deck of the Evo for 8 hours after spraying down all its surfaces with 70% ethanol. We can see that there's this single contamination event being recorded. At the same time, we added TSA and SAB agar settle plates that were left on the Ticon Evo deck for the same 8-hour period, and they showed no signs of contamination or growth after incubation. These results, combined with the contamination previously seen, showed us that it was likely due to the use of these standard tips with the device that led to the contamination events. We actually blamed our colleague Sten for this single contamination event seen. Joking, not joking. So in answer to this question that we posed, we saw no contamination on the settle plates on deck after 8 hours in the open lab. That single contamination event in the manually dispensed plate meant that things were not conclusive, and we'd like to run this again to see if it may have been something to do with the preparation of the plate rather than it being on deck for 8 hours. But we did conclude that filter tips are a must to prevent contamination of media when using an automated device in the lab. We hypothesized that the filter tip was giving us protection from either aerosol formation or from contamination from the system liquid from the device that it uses to aspirate instead of air. Over the years, we've run quite a few complex multivariate experiments using automation at Synthace, and we've always been curious to the extent that evaporation may make our solute concentrations in the wells differ from what's expected. So here we turn our attention to studying evaporation during automated experiments. At first, we wondered if we could use the literature formula for evaporation to see what extent evaporation might occur during an experiment. And here it is. I won't go into it because Chris did the work, but I wanted to mention that we measure these things during an experiment to get a model of evaporation rate and its impact on the volume inside a well. For instance, the velocity of air above the water surface was measured using a anemometer, and this was set up above the plate. So we do get the data for it, plug it in and see how it impacts, what it predicts. So the experiment we ran, we wanted to evaluate things that we thought would impact evaporation as well. So we could predict, using the literature formula, as well as measure and compare. So these are the experiment factors we investigated during this experiment. So we varied the surface area by using either 96 or 384 well plate. We varied the initial volume in each of the well types. We changed the total moles of solute that the liquid was comprised of, and also its composition, making mixtures of either DMSO, sodium chloride, and glycerol, mixtures of those three things. We measured this thing over time, over 18 hours, and we also monitored humidity, temperature, and pressure. And after setting up this plate, we then moved it to the plate reader and measured the liquid height every hour for the duration of the experiment. These were the results of the experiment. On the left, you can see the model calculation of the liquid volume in the well over time. And on the right, you can see the measured results of the liquid volume calculated from the measure of liquid height and knowing the geometry of the well. The results show how the model overestimated the impact of evaporation on the well compared to the measured results. So at least things aren't as bad as what the literature thinks. Because here it's saying if we start with a total volume of 100 microliters in the well, we can expect it to be completely evaporated after 18 hours. But this wasn't the case. So that's good news. The measurements that showed, however, that the evaporation of between 10 to 20% of the initial total volume of the well occurred over the 18 hours of the experiment. So this is quite impactful on the concentration of our solutes compared to what we have actually targeted. So something to bear in mind. This analysis shows the change in measured liquid height over time. So this how the liquid's decreasing over time as a measure of the impact on evaporation and the effects of solute concentration as well as the initial volume, liquid volume in the well. And it was a little bit counterintuitive to what our initial understanding was, that the lower the initial volume in the well, we actually get slower rates of evaporation. So that was surprising. So in answering the question, can we use the literature model of evaporation to predict its impact during the experiment? The answer is no, but we can augment the model to include the initial liquid height, the well surface area, and the volume fraction of water to create a model that can make some accurate predictions of evaporation over time. Let's now move on to an example of an assay where we noticed that evaporation over time was actually a problem. This relates to an automated qPCR protocol discussed earlier. As mentioned, we were optimizing the liquid classes for handling both DNA and master mixture in the assay. And for the experiments, we used the same stock of DNA at a set concentration. Shown in the graph is the CT values that are measured during the qPCR assay. The lower the CT value, the higher the amount of DNA is present in the well. And we saw this sort of systematic trend over time of increasing measured DNA concentrations. And our hypothesis was that this may be caused due to concentrating of the DNA because of evaporation. This is the flow of the qPCR assay. The DNA and master mix stocks are in a source plate. They are then combined and diluted on a dilution plate before then dispensing the final reaction volumes to a plate before measuring it on the qPCR device. So we wanted to test the evaporation hypothesis, seeing if this was either occurring due to evaporations of the stocks in the source plate or the final reactions in the final plate, possibly both. We thought the effect leading to the concentrating of the DNA over time was independent of the automation preparation. And we showed this by running the assay using the Dragonfly, which is very rapid, completing this assay, the same assay, in 4 minutes. Here is the CT data and showing that the effect wasn't impacted by the use of automated liquid handling, but was more time dependent. So this is why we thought to look at the stocks. The next steps were to assess where evaporation was impacting the assay. the DNA and the master mix stocks were dispensed to a source plate, and a set of reactions were set up representing time zero for the evaluation. The time zero reactions were then aliquoted to the final plate in replicates, with some being covered and some remaining open. Then the same stocks in different source plates were subject to different treatments, where the stocks were either kept at room temperature or incubated on ice, as well as either being open or covered. And these conditions were left for 30 minutes. After 30 minutes, these samples that were then placed on the final reaction plate before measuring using the qPCR device. Looking at the reactions from time zero, we can see a negligible difference between sealed and unsealed plates for the final plate. So that's good, and maybe our focus should be about the initial source plate. We saw a concentration of DNA for the stocks incubated at room temperature and unsealed for 30 minutes. So this is after 30 minutes, unsealed and at room temperature, we see a concentration of the DNA stocks. This is partially recovered back to the original stock concentrations by sealing the plate. And then if this is just put on ice without any sealing, we also get a partial recovery. But it's when we combine on ice and sealing the plate that we see comparable levels to the original CT values. So in order to prevent evaporation effect leading to a systematic error on the qPCR assay, we recommend keeping stocks cold and sealed during the protocol, or indeed splitting the amounts of runs up and executing the assay as quickly as possible to prevent this effect. This is just a quick note about sample stability. It may be necessary to keep liquids cool to prevent their degradation, but remember this may increase their viscosity and you may have to adjust your liquid classes accordingly. Moreover, if you can make the execution of your automated device as quick as possible by ensuring things like your multi-channeling, or indeed by using something like a rapid dispenser, then you can limit the degradation of your samples. This is the final section now, and before we head to the Q&A, I wanted to show you some capabilities of automated devices that can help whilst handling biological samples. The first thing to look at is adding peripheral devices that can increase the capabilities of your liquid handling device. You can set something up like a plate washer that's accessible to the robot arm of this liquid handling device. The peripheral device is often quite specialized, and I'm showing a plate washer which can do things like buffer switching, cell washing, shaking and soaking, and it works very efficiently during an automated protocol. And this is instead of trying to program the liquid handler to perform a cell wash that the plate washer can do quicker and with less consumables. We can also use sensors incorporated into the automated devices to help with transferring biological samples or reagents. This is from Hamilton, where there is a pressure sensor monitoring the pressure in each of the pipette channels during aspiration and dispensing steps in real time. And you can see this profile that you generate from an aspiration event. The plunger moves up, pressure decreases, push out, and it equalizes. You can also get the same profile for dispensing. And where it becomes pretty cool is you can use these profiles to get things like clots in the sample. If you're pipetting blood and you can create this profile in green for a successful aspiration of the sample, then you can set thresholds on performance for that transfer. Ooh. The blue shows the decreased pressure in the tip due to a clot preventing aspiration of the total sample. And this will then automatically mark that pipetting step as failed and alert you to this. This is pretty cool. You can also use it for detecting insufficient liquids from the source, so you get a different profile. As well as you can see if foam has been aspirated, just based on the profiles that you get. So it's liquid agnostic. And finally, I just wanted to show you something else that I thought was cool. This is from the Echo, and it has the capability called dynamic fluid analysis. This transducer, it's one well to one well, and it uses acoustics to generate these droplets that go up and stick to the plate, and it's 2.5 nanoliters to 25 nanoliters. This transducer can also measure the echoes that come back from this transfer, and it can detect where the bottom of the source and destination plate wells are, as well as where the liquid meniscus is. So it gets an idea of exactly what's going on in each transfer, and it can adjust the power it gives to give you the correct transfer of liquid that's required, so the desired volume, and then you can apply this to later experiments. So it almost does automatic liquid class optimization for you. I haven't really had much experience with this, but I think it's, yeah, pretty cool. And finally, I'd just like to say thank you to Daniel, Nathan, Anne, and Luke for the webinar for the series so far, and Sten, Fatima, Lucy, Nuno, and Chris, as well as Marcus for the data and help with putting the presentation together. With that, I'll open up the Q&A. Thank you very much.
Thank you very much, Steve. So yeah, that was a fantastic coverage of all sorts of different subjects, which can make dealing biological samples a little bit challenging, but possible to overcome. And yeah, we've got a few minutes left at the end of the webinar to tackle a few questions. Our first question is one that's, you've gone through a lot of different automation here and different automation strategies. So the question is, have you found any hardware that's particularly good at transferring mammalian cells? Yeah, if that's something that either you or Nuno, who's also joined us for this question session, would like to take a stab at.
Yeah, maybe I can start, give Steve a couple minutes to recover. So I think that's a really good question, and I think there's a very reassuring answer. And Steve illustrated that throughout his talk. I think most devices can be adapted to deal with the complexity of, say, mammalian cells. And that could be as simple as adapting your experiment conditions slightly, so you don't even need to necessarily change the device in any way. But for example, you might change the concentration of your source of cells, for example, so that you're pipetting more accurately. Perhaps changing things as the tip type, make sure you're using filter tips and whiteboard tips instead of just the standard tips that perhaps you've been using so far. Obviously, if you have an unlimited budget, again, I wouldn't necessarily consider only what is the best device to pipette mammalian cells, because perhaps I'm not even sure if there has been a proper direct comparison of all these different devices, but rather thinking of how are you using these cells? What is the best device for the type of experiments that you're running? As Steve pointed out, a plate washer can be incredibly useful, and perhaps a plate washer in combination with a dispenser is a really good combination. But if you then also need to do a serial dilution, then a liquid handler would be great. Or for example, if you already have a hood and you need to make sure that the device you're using fits in that hood, perhaps, and your actual limitation is just the size of the device. And yeah, there are a few dispensers that are smaller, have a smaller footprint that can fit. But again, we've shown that you can use a big liquid handler in the lab that is handling bacterial cells as well. And as long as you keep things clean, you're using filter tips, you can very easily use that device as well.
Yeah, I think this is maybe one of the most surprising results that we've had in the lab, actually, because there tends to be this sense that cells will get contaminated at the drop of a hat, but it's my suspicion that actually it's mostly the scientists themselves that end up contaminating things. So if you're using automation, then it can reduce that chance of contamination in the first place. But yeah, I guess so what Nuno's saying there is it isn't so much the bit of hardware that you're using, it's more like all of the different measures that you can take to try and then make sure that those cells are transferred successfully. And as Steve covered in the talk, that's both about making sure that they don't settle out, but also about making sure that they're not subjected to undue shear forces. Those seem to be the key factors that you need to control, as well as sterility. Steve, have you got any more you want to chat about on that?
No, I think you've covered it. I was going to suggest a cheat answer that I like the Amber 250 for handling mammalian cells in an automated way, but I don't think that's quite what the question was looking for.
Yeah, if for those of you who aren't aware, the Amber 250 is a bioreactor system which can culture cells up to 250 milliliters, hence its name. But yeah, not so much for just standard liquid handling. Might be a little bit overkill. We've got a few more questions here, but honestly, it's quite hard to choose between them. So I think for the purposes of today, we'll wrap it up here. Thank you very much to Steve and to Nuno briefly for jumping on, helping us handle questions. And it just remains for me to talk about our next webinar, which is going to be on December the 6th, where I'm going to be discussing how to run powerful automated DOEs. If you've missed any parts of today's masterclass, and there was a lot of information in there, a lot of graphs that you might want to go back and have a look at, don't worry, we'll be sending you today's recording along with all other related content. If you've got any more questions, then get in touch with us at hello@synthace.com. And those of you who've asked questions that we haven't been able to get to, then we'll get in touch with you to answer those after the webinar. So thank you very much to Steve for a fantastic talk. Thanks to Nuno for jumping in questions. And thank you all for attending and listening. Thank you very much.
Bye, everyone. Bye.